In the 1850s, a Swiss biologist, Albert von Kolliker, was looking at muscle cells and noticed that there was lots and lots of these little granules. Later they would come to be known as mitochondria. And can you guess what they do?
Ooh, I know this one.
Yes, Bartleby. Mitochondria are the powerhouse of the cell. Yep, and that's pretty much all you need to know about them. Not really. There's lots more. Let's see. If you remember from the endosymbiosis lecture, mitochondria are basically these captured bacteria. Some bacteria today still operate this way in terms of making energy. But the deal that we and pretty much all other eukaryotes made with them is we'll give you food and oxygen and you give us lots of ATP. So oxygen's easy enough as long as we're breathing in and out. We should be supplying them with oxygen. Food, it's a little bit trickier. We can't just hand them a glucose. We have to kind of turn it into little mitochondria baby food and at least cut it in half and feed it to them. They need smaller molecules. Usually start with kind of a 3-carbon molecule called pyruvate. Now inside mitochondria, they look a little bit more complicated, right? There's all these folded membranes, and those are going to be important for the chemical reactions that happen inside there. Well, in the mitochondria, there's 2 sets of chemical reactions, citric acid cycle and the electron transport system, that further extract energy from pyruvate. When we look at this energy diagram, we ended off with glycolysis with a couple of pyruvates left over from that glucose. And in this case, we're going to break those down into 3 carbon dioxide molecules, and we're going to extract energy from that. So you said before that energy takes a lot of forms, so which kind is this? Yeah, I was just about to get to that, Bartleby. I think he's really hooked. Now he thinks it's more than just the powerhouse of the cell. Yeah, it's actually kind of interesting. Plus, you're a really good teacher. Well, thank you, that's very kind. I think you might be a little biased. Anyway, we're going to extract energy. Got to pay thermodynamics, so of course some of that energy is just heat energy. Also, we're going to get just a little ATP out of it. I know we're still waiting for the big payoff. That's this one, the electron transport system. So a lot of the energy that comes out is actually stored in this guy, NADH, one of those electron carriers. And we even had some left over from glycolysis. That's what we're going to hand off to the electron transport system to do the rest of the work for us. Let's take a look at where these take place in the mitochondria and then jump into the citric acid cycle. You can see that there are 2 sets of membranes, the folded up one in the middle and that outer membrane. The citric acid cycle takes place all the way on the inside in the matrix, this kind of blue area. That's where those chemical reactions take place, breaking down pyruvate. And the electron transport system takes place in this inner membrane. So right there on that kind of yellow line, there's a bunch of proteins embedded in that membrane that are going to drive the electron transport system. Well, here's the citric acid cycle. Looks a lot like glycolysis, doesn't it? Lots of chemicals, lots of arrows. Well, let's take it one piece at a time. First, let me start by saying it's also known as the Krebs cycle or even the tricarboxylic acid cycle. So you might hear it by a few names. I'm going to stick with citric acid cycle. Starts with citrate or citric acid. And then there's lots of different chemical reactions that then end with citrate. And before we do that, there's this preparation step where we convert pyruvate into acetyl-CoA. So that's not really part of the cycle. I think just for our purposes, if we just include all of this under the banner of citric acid cycle, it makes it a little bit simpler, even if it's not entirely accurate. All right, so first, on our first pass, I want to track the carbons, basically the food molecules. This started out as glucose, and through glycolysis, I got a couple of pyruvates. So let's start there with this 3-carbon molecule, pyruvate, and see how long it takes us to break it down. Well, in this very first reaction, I give off carbon dioxide. So there's one of the 3 carbons broken down. So now I just have this 2-carbon molecule left. That gets passed off and joined to that oxaloacetate to become a 6-carbon citrate. Now we're moving around. Oh, there's my other carbon dioxide. So there's 2 of the 3 gone. And then I keep going around, and there's my third one. All 3 of the pyruvate molecules have now left this cycle. And then they're going to diffuse out of the cell, and we exhale them, which really means that the food molecule that came in, whether that came from a soda or a sandwich, that glucose got chopped up, became pyruvate. Now it's all carbon dioxide, and you exhale your food. Kind of a strange idea, but there we go. Moving around the rest of this cycle is all about just more energy transfers and basically completing the reaction. But your carbons are gone. Let's take another pass, this time looking for ATP, our main energy molecule that we're after. We'll start up here again with pyruvate. Nothing yet. Still just breaking down molecules, breaking down molecules, breaking down molecules. Oh, I see a phosphate. There's our ATP. Looks like first we get GTP, and then that phosphate gets transferred to ADP. So I got 1 ATP, and if you wanted to think of it per glucose, we'd multiply it by 2 since we started with 2 pyruvates. But there's the ATP. Is there any other ATP? Nope, that's it. Not a big payoff. So it looks like for every glucose, I got 2 ATP from glycolysis and 2 from the citric acid cycle. Where's the rest of my energy? Well, let's see if we can find the rest of the energy in the electron carriers. And those are going to be our NADH and FADH2 molecules. You can see them highlighted there in orange. But let's just go around and track it again from pyruvate. Here we catch 1 NADH from that very first transfer. Kind of makes sense. We're breaking one of these carbon-carbon bonds to give off carbon dioxide. So we should extract some energy from that. And we'll carry on from there, moving around. And as we break that molecule down, we're breaking more bonds. There's a second NADH. Keep breaking bonds and give off more energy. There's a third NADH. Making our way around. Some is ATP. And here's another one, FADH2. That's another electron carrier. And then finally, a 5th electron carrier. All right, so we have a lot of electrons being held by these electron carriers. Obviously, that's going to go into the electron transport system. All right, so we're done with the citric acid cycle, but we don't have a whole lot more ATP to show for it. The big payoff comes in this last step, the electron transport system. And there's really 2 parts to this, so don't make it too confusing, just really the 2 main parts. First, we build up this hydrogen ion gradient. Oh, gradients again? I know, more gradients. But out here, we're going to use the power of those electrons to build up a hydrogen ion concentration. And then we're going to synthesize ATP by letting that hydrogen come back in through this special protein called ATP synthase that obviously makes ATP. Let's take a closer look. So here we are in the mitochondria. There's that outer membrane, and here's that inner membrane, the one that's all folded up, which contains lots of these proteins shown in blue here. So that's where the action is. One thing I do want to note is there are quite a few other terms down here at the bottom that we can throw around at this. If you're in my class, I'm just going to use electron transport system for the whole thing. It just is a little bit simpler. The names are not as important to me, more the concept of what's going on here. So let's pick it up from where we left off. The citric acid cycle gave us lots of electron carriers, this NADH. We also have some of that from glycolysis. We also have FADH2. So their job is to be electron carriers, right? They have been reduced, and now we're going to oxidize them and give up those electrons. So then we end up with the oxidized form of this that's going to go back to the citric acid cycle, get charged up with more electrons, and come back to here. All right, so there's that part. We got these electrons. Obviously, these have energy, and they're going to power this protein right here, and it's going to pump a hydrogen ion across the membrane. All right, maybe several of them even. The electrons keep flowing from one molecule to the next, and we get, in this protein complex, more hydrogen ions being pumped across. The electrons keep moving through, and we get more hydrogen ions pumped across. So that's stage one. We have created a hydrogen ion gradient. Lots and lots of hydrogen ions, which means it's pretty acidic. It's got a low pH out here in this intermembrane space. And these hydrogen ions want to come back in and flow down their concentration gradient. But remember, this phospholipid bilayer is a barrier, so they can't. But we're going to allow them to come through this molecule. ATP synthase has a pretty obvious job, doesn't it? It synthesizes ATP. And so as the hydrogen ions come through, there's actually like a little motor that spins around, and as it does, it puts together an ADP with another phosphate, creating ATP. As long as hydrogen ions are flowing through, then it keeps creating more and more ATP. And not just a little bit of it. Remember, we had a lot of these electron carriers, so there's a lot of power there. And this creates somewhere around 32 ATP molecules, and that's per glucose that we looked at. So we got 2 from glycolysis, 2 from citric acid cycle, and now 32 from the electron transport system. That's a big payoff. That's what we've been waiting for. Before we leave the electron transport system, there's one more item to deal with, and it's an important one. Oxygen. Yeah, I knew I needed that for something. It's the electron acceptor. So here are those electrons. They were dropped off by NADH and our other electron carriers, and they flowed through, doing a lot of work, creating a hydrogen ion gradient, but they can't just be let loose. That would cause a lot of destruction. So oxygen accepts them, combines them with some hydrogens, and becomes water, threat neutralized. And this supplies about 10% of our water needs each day. It's a nice bonus. Another little interesting side note is that quite a few of these molecules, these proteins that pass electrons like cytochrome C and others, and even ATP synthase, these are the targets of several types of poisons like fungicides and insecticides. Even cyanide targets this one right here, and it blocks them up. And so you could imagine that with these not working within mitochondria, how that could cause a real problem in terms of your ability to produce energy to function. Well, obviously mitochondria are vital to our cells because of the amount of ATP they produce. It dwarfs the just 2 ATP we got out of glycolysis that were done in the cytoplasm. These 2 sets of chemical reactions within the mitochondria produce a lot more ATP for us. And most cells really depend on that. There's only a few that get by without mitochondria. When you look at these complex chemical reactions, make sure you're focusing on the key points. They can be overwhelming to look at first. But I would say one thing to focus on is looking at how the carbon molecules, the food molecules, are broken down. Usually it's in a lot of different steps, just slowly extracting those electrons from the food molecule. And then 2, where's the energy coming from? How is it leaving each of those chemical reactions? We're using one chemical reaction to drive another one. The big point is to drive the chemical reaction that makes ATP to power our cells. And then once it's used, it makes ADP, and we keep recycling that again and again. Well, that's it for this time. Until next time, keep appreciating life, especially your friends, the mitochondria. Keep them happy.